very distinguished scientist, Dr. Viratmat sir, Dr. Krishna Kumar sir, uh, all other distinguished scientists, and my fellow speakers, other participating scientists, and if there are any students. I am very pleased to be a part of this very important and relevant discussion meeting. I'll just straight come to the title, as Dr. Krishna Kumar has announced, is Molecular Mechanisms Underlying Symptom Development in Phytoplasma Associated Diseases, the Key Players and Their Role. So this presentation is different from most of the participations, most of the presentations which were going on so far. I am from Department of Botany, University of Delhi, and I will be skipping some of my introductory initial slides because they are very basic information about phytoplasma and they have been already deliberated enough, except for just to build up the tempo that phytoplasma has many unique features, though they are bacteria, but because of the fact that they lack cell wall and their genome size is intriguingly small and they have a very low GC content and highly reduced genome, and despite being a prokaryote, uh, unexpectedly its repetitive DNA content is very high. And again, unlike other molecules, it uses UGA as the stop codon, as culturability of it is a disputed, a contentious issue, and most importantly, it has a trans-kingdom existence. Economic importance of phytoplasma, again, I would not like to say much, but since we have been since morning portraying phytoplasma in a very villainish way, I think it's in a lighter way that it's an irony that phytoplasma is a cause of the ornamental value of a very beautiful ornamental plant, poinsettia. The branching poinsettias and the green chrysanthemums and some of the exotic flowers, they have their ornamental value enhanced because of the presence of phytoplasma, and that itself is actually one of the quarantine concern, which has been very rightly and very emphatically pointed out by my earlier speakers. Again, I will skip these slides because we have heard enough about these things. Uh, since I am focusing mostly on the symptoms and their development, there are varied, vast array of symptoms which are produced by phytoplasma association in the host taxa, which vary from which is bloom to virescence or little leaf or yellowing or stunting. But coming from a plant biology background or a basic botany background, I would like to elaborate upon these symptoms, or I look at these symptoms from a botanist angle. So when a plant pathologist calls a symptom as a witch's broom, a botanist actually looks at it as shortening of internodes and axillary shoot outgrowth. When a plant pathologist calls a flower as virescent flowers, we call it as greening of the floral whorls. Or when a plant pathologist calls it a fasciation, for us, it's a very intriguing symptom where a stem, which is normally a round stem, it becomes dorso-ventrally flattened, which is an exception in the case of angiosperms. Similarly, when we say phyllodes, normally a flower is a determinate structure. When a plant decides to flower, that apical meristem becomes determinate, and in case of phyllodes, that determinacy or that determinate behavior is absolutely ransacked by the phytoplasma and it becomes indeterminate. And interestingly, from within the flowers, the shoots start emerging, and all these features are the factors which are responsible for a person like me, who is neither a pathologist nor an entomologist, to be interested in the development of these symptoms as a plant biologist. In fact, I always like to show this slide. It's, as has been already said enough, that till 2009, nothing like publications in the field of phytoplasma research were not too many. It is this paper in 2009 which said phytoplasma research begins to bloom, which came in 2009. And since then, it is not only blooming, I would say there is an explosion of publications in phytoplasma, which is a very satisfying thing to all phytoplasma biologists. My group in botany department, Delhi University, has been working on phytoplasma, I think some years even before 2009. So starting from reporting new phytoplasma groups, evolutionary lineages within the phytoplasma group, these are the various work which we have been doing, with a very special focus on sesame, because sesame is one of the worst affected crop plant, at least in India, and it's an important oilseed crop, but it is very badly affected by phytoplasma. And these are the different stages how phytoplasma affects causes or is associated with phyllodes in sesame. So we have been actually surveying throughout the country all the sesame growing states from the different parts of the country, collecting it and detecting and characterizing and ascertaining the phylogenetic relationships between the phytoplasma groups which affect the sesame crop. Here, sir, I would like to take 2 minutes. I was very happy when Dr. Viraktamat, very, very highly respected, he's a teacher of our teachers actually, pointed out that there is a need to do, quantitation or estimation of the phytoplasma titer in different parts of the plant. Sir, this is the work which is already published in 2017, where we took sesame plants and from different parts of the plant during four different stages of the disease progression, using qPCR method, we had quantified the phytoplasma titer not only in different parts of the plant but also during disease progression. As you very rightly said, the range of phytoplasma titer in different parts of the plant and during disease progression is really surprisingly wide. So this was a very good result we got, and we said that this is a very important information for not only for effective sampling of the disease material, but also for understanding how disease progresses in phytoplasma affected plants. Sir, am I audible? Can you all hear me? Okay, sir. Okay. So another thing which, not only that, we are also trying to understand and complete, how it hijacks the entire development of the plant. And here I would like to, because there was a question on pollen grains by somebody. So we said we had, in fact, done a very detailed investigation on every part of the phytoplasma affected plant, and not only each whorl of the flower, each part of the flower, including even pollen grains. And if you can see it very clearly that these are the healthy pollen and these are the phytoplasma. I mean, mostly the flowers are not either formed. And a very important thing is that stamens are the last whorl to get badly affected. In fact, we were thinking for a very long time that stamens do not get affected, but finally if the disease becomes very aggressive, even the stamens become become defective, and the pollen grains show very clearly abnormal development. And we have even done the staining by fluorescent dyes, and we saw that pollen is not viable if it is a phytoplasma affected. At least in sesame I can say that, sir. Okay, so coming to the precise topic of my talk, mechanism of symptom development, it is very intriguing and very important to delineate, not only to understand or to develop the control measures of phytoplasma, but also to understand the plant development biology, because this is a very unconventional approach to understand how different plant parts develop and how their development can get affected by biotic stresses. So this is the way I was telling you that we have been analyzing every part of sesame plant and every whirl of even the sesame flower, how it gets affected by phytoplasma. So essentially what we can conclude and what even literature agrees is that whatever changes are caused, basically they are developmental alterations. So it is the development, to be very precise, in not in all cases I would say, but most of the cases it is the development of the plant which gets altered. So so if we have to go further, we have to understand that though we know that vector brings the phytoplasma to the host plant, and the fact remains that if it is the development which is mainly the target of the symptoms, we know that phytoplasma remains in the phloem. That means it remains in the tracheary elements or in the vasculature. And it's a very well-known fact that the development of the plant is basically governed by this region, which is the shoot apical meristem. All the genes, all the molecular mechanisms which have been deciphered in the plant development, actually they are very active in this shoot apical region. And it's very important and interesting to point out that phytoplasma never reaches here. It is vasculature limited, so it never can actually physically reach the niche which is involved in the development. So that has led to the discovery of what we call as the effector molecules, because this is the region, all the pathways, clavata pathway, Bushel pathway, which have been discovered in the plant development, they operate in this very dome, whereas the phytoplasma is here. That means it is secreting some substances which leave its natural niche, which is phloem, and then after traveling through the cells, it reaches this part of the SAM and then hijacks the development program of the plant. So this is what brings us to the study of effectors, and we have been studying effectors of phytoplasma for past some time. Before I reach to my studies, I would just like to outline that, if we say specifically talk about phyllodes or any symptom which affects the flowering of the plant, we have to understand in the normal cases, there is a shoot apical meristem which is vegetative, and there comes a time when the meristem identity changes and it becomes the inflorescence meristem, and there are inflorescence meristem identity genes which have been identified. Then there comes a time then this inflorescence becomes floral meristem, and we have very well, ABC model and all those similar studies are there which have enabled us to understand floral meristem identity genes. And then come the floral organ genes, that means the floral organ identity genes. If this is the scheme of a normal flowering, and if phytoplasma is able to genetically disrupt all these processes, then definitely it has to be understood or assumed that these effectors, which are actually released by the phytoplasma, are targeting either some of these genes or all of these genes fully or partially. So if we have to understand the molecular mechanism of symptom development, we have to be actually working in this area.